Good afternoon and welcome back everybody for our short talk session. So I'm absolutely thrilled this afternoon to be back. I'm supposed to be David, so I've had some therapeutics myself clearly in the coffee break. And this afternoon we've got 3 presentations from researchers working with UK Spine, and they really are super projects. First up, I'm going to hand you over to Satomi Miwa, who is over at the University of Newcastle. And I hope the sun is also shining in Newcastle today.
Yes, indeed.
And Satomi is going to present on preclinical validation of novel candidate senolytics. And this is a really lovely project that Satomi's been working on through UK Spine for about 1.5 years now, isn't that right, Satomi?
Yes, that's true. Yeah.
So the floor is yours.
Okay, thank you very much for the opportunity for me to give a talk. The title of my talk is Preclinical Validation of Novel Candidate Senolytics. And senolytics come from 2 words, senescence cells and lysis. So what is senescence cells? Different type of cells become senesced state by telomere attrition, DNA damage, and stresses, different kinds of stress such as oxidative or oncogene. Senescence cells are different. They tend to become larger, nuclear size enlarge. They release pro-inflammatory cytokines and reactive oxygen species, have mitochondrial dysfunction, and metabolic changes. We looked at mitochondrial function during the development of senescence from here by damage-induced senescence. As you can see, green line is respiratory control ratio, it's mitochondrial coupling, rapidly deteriorate quite fast after a damage, whereas loss production goes up more gradually with time. The problem of senescence cell is that because of these secretory factors, they can influence surrounding cells to become senesced. It's called bystander effect. And the senescence cells spread. Now, recently, the approaches to selectively target senescent cells, developed called senolytics. And it's been shown in mouse models. These approaches are able to prevent or improve many age-related conditions, such as muscle weakness, cataract, lipodystrophy, atherosclerosis, pulmonary fibrosis, osteosis, osteoarthritis, osteoporosis, and many more. And typically, senolytics target anti-apoptosis pathway. For example, BCL2 inhibitors, such as navitoclax, are senolytics. However, because in nature they are cytotoxic, they do have side effects. So our aim is to develop safer and more efficient senolytics. Of course, there are other groups in the world who are interested in this and doing high-throughput screening, bioinformatics. However, we had an instinct-educated guess to test. We hypothesized that because mitochondrial senescence cells has distinct problems, if we target them along BCL2 inhibitors, we might be able to enhance apoptosis activity and hence improve sense specificity and the efficacy of senolytics. So we wanted to test combination of navitoclax, a BCL2 inhibitor senolytic, plus metabolic targeting drug. I'm sorry, I cannot say too much more exactly what it is for the moment. And we wanted to test both in vitro and in vivo settings. And the in vivo part is the one supported by UK Spine, preclinical validation of novel candidate senolytics. Firstly, in vivo test, we co-cultured fluorescence labeled young and senescent cells, green senescent cells, and red are young cells. We treated the cells with a test drug with a range of concentrations for 3 days, and then counted the surviving cells and plotted the results in a graph. So as you can see, with 10 micromolar navitoclax, after 3 days, you see much less green cells are remaining. Now this is our new results. Y-axis cell number, percent of baseline, and then this is after 3 days. Light blue are young cells. Light orange is senescent cells treated with increased dose of nabitoclax. And senescent cells start dying with increasing nabitoclax, whereas young cell, light blue, relatively unaffected. Now, purple line shows young cells treated with nabitoclax plus metabolic targeting drug. Essentially, there's no additional effects. However, dark orange, which is senescent cells treated with nabitoclax plus metabolic targeting drug. As you can see, already these very low nabitoclax concentrations, senescent cells start dying, whereas young cells totally unaffected. From here, we can show with combination treatment, we can increase specificity of senolytic action just to senescent cells, and also increase efficacy of killing senescent cells. So our hypothesis has been shown, proven true in vitro. How about in vivo? We used a mouse model of premature aging by radiotherapy. We are interested in this model because about 2 million people, long-term cancer survivors in UK, suffer from premature aging, and there are currently no effective treatment. It is hypothesized that this is caused by DNA damaging therapies such as radiotherapy or chemotherapy, making a lot of senescent cells. So this is what we did in mice. We irradiated mice, radiotherapy, whole body irradiation at 5 months old. And once they are recovered after 1 month, we gave treatment. And then we followed up for aging phenotype. We had 5 groups, controls, low nabitoclax. We used 100 of typically used doses in literature. We had to make an educated guess to decide this dose. It's very low. Metabolic drug alone, and standard nabitoclax dose, and combination, low dose, this dose of nabitoclax plus metabolic drug. Now, this is a frailty score. Baseline before irradiation, 5 groups. The mice have been assigned randomly to 5 groups. So there's no group difference. And we gave irradiation. As you can see, frailty score increased after irradiation, but there's no differences amongst groups. Of course not. And then we gave treatment here. As you can see, after only 1.5 months, we can see control group further increase frailty score. However, both a standard dose of nabitoclax and combination therapy are able to suppress increase in frailty score. We also looked at cognitive function by Y-maze test, looking at spontaneous alteration, short-term memory test. Now it's higher the score, the better. You can see standard nabitoclax dose, this one, and the combination therapy outperforms other groups, especially the controls. Now, neuromuscular function, which is hanging wire performance, again, standard nabitoclax group and combination group outperforms other groups. And here, again, I say, this is a combination of this low nabitoclax and metabolic drug. With a single treatment, they do no better than control. However, in combination, they do as good as very high dose of nabitoclax. To summarize, in support of our hypothesis, metabolic targeting drug can sensitize the action of senolytics and reduces the dose required. We have shown only 100 needed, so it's safer. In addition, from this project, we established both in vitro and in vivo testing platform for senolytics, if anybody is interested. We saw combination idea can be applied to other anti-apoptosis targeting senolytics, which we have been testing, and also cancer drugs. And cancer cells also have mitochondrial abnormalities. And finally, this study is still ongoing. We are looking at long-term benefit of this treatment. And if anybody is interested in looking at our mice tissues, biomarkers, please let us know. An acknowledgement, thank you so much for Thomas Bonds-Gretzky, my mentor, and people in our group, especially Edward Fielder did a lot of experiments presented in talk, along Abbas Ishak, who did a lot of mouse experiments. And there's a wonderful teams. And thank you to our funders, especially UK Spine, for allowing us to do these very exciting experiments. And thank you very much.
Thank you very much, Saitomi. It's lovely to hear your presentation and the progress that you're making with your project. In the interest of time, we're going to move on and do all of the talks this afternoon and come back if we've got time for questions. But please do post questions in the chat if you have any. So next up, I'm just checking the running order. Is it Helen that we've got next up? Helen.
Hello.
Hello. So joining us from Duncan Richards' group, we have Helen Knowles, who is going to be talking about one of our other proof of concept projects around using oxygen nanobubbles. And I don't want to steal any of your thunder, Helen, so I'm just going to hand straight over to you. Please take the floor.
Okay. Right. Hopefully that looks okay. So the work I'm discussing today forms part of a project based at the University of Oxford, where we aim to identify a translational pathway to address tissue hypoxia and its consequences using novel oxygen nanobubble delivery technology. In a slightly backwards acknowledgement slide, the people listed here, all the people involved in the project. So we are using orally delivered lipid-coated oxygen-loaded nanobubbles. So there's an image shown on the right-hand side here to see what they look like. Lecithin is the main lipid component of these nanobubbles, and it's mixed with glyceric acid, glycerol, and citric acid. And in this solution, the lecithin component naturally forms these nanobubble-like structures, which can then be loaded with oxygen by bubbling the gas through the solution. Professor Eleanor Stride's group have shown that these nanobubbles can be used as an oxygen delivery system to reduce tissue hypoxia. So I'm just going to briefly go over two studies they have already performed. So there's a cancer therapy study. They used a mouse pancreatic cancer xenograft model, and 30 minutes post-oral gavage with the oxygen-loaded nanobubbles, they saw reduced tumour expression of the hypoxia-inducible transcription factor, HIF1 alpha, as well as other hypoxia markers such as vascular endothelial growth factor. So if you look at the graph on the right here, compared with oxygen-saturated water, the mice that were treated with the oxygen-loaded nanobubbles, you saw significantly reduced expression of HIF1 alpha protein in the tumour tissue. They also used an argon-loaded nanobubble mix, which also reduced expression of HIF, although not to the degree that the oxygen-loaded nanobubbles did, suggesting that there might be some element of effect of the nanobubbles themselves, but an additional component that's dependent on oxygen. They've also performed an exercise performance study where 10 competitive male cyclists were given oxygen nanobubble drink and then asked to perform a 16-kilometre time trial. And those cyclists that received the oxygen nanobubble drink compared with the placebo control exhibited a greater power output during the trial, as well as reduced time to completion of the trial. So this study uses rheumatoid arthritis as a test system in which to assess the ability of the oxygen-loaded nanobubbles to reduce hypoxia in a disease situation. It's well known that hypoxia affects key disease mechanisms in rheumatoid arthritis, including inflammation, angiogenesis, cartilage destruction, and bone erosion. And it's equally well known that the hypoxia-inducible transcription factor HIF induces expression of many cytokines, chemokines, and other factors that drive these processes. So there are two objectives to the overall project. First objective is to look at the in vitro cell biology associated with improved oxygen delivery by the oxygen-loaded nanobubbles. And this involves looking at individual cell types in vitro that are associated with the rheumatoid joint. So osteoclasts, T cells, fibroblasts, endothelial cells, etc. The second objective is a clinical study looking at oral delivery of oxygen-loaded nanobubbles to rheumatoid arthritis patients, and then looking to see if there is any effect of this intervention on HIF marker proteins and also on hypoxia-affected disease pathways. However, the talk today focuses on one component of Objective 1. So we're looking at osteoclasts, which are the primary cell type that drives bone erosion in the rheumatoid joint, as well as the monocytes from which they derive. So we form primary human osteoclasts in vitro from peripheral human blood, which we obtain as leukocyte cones from the blood transfusion service. We positively select CD14 positive monocytes, which we then treat with MCSF and rank ligand, which are osteoclastogenic cytokines, for 9 days. This treatment induces fusion of the monocytes to form active mature osteoclasts. If you look at the images on the bottom right here, osteoclasts are defined as TRAC-positive cells with 3 or more nuclei, and if you grow them on bone, they digest the bone, and there's bone resorption. You can see the purple resorption tracks here. So we have looked at the effects of nanobubbles loaded with 3 different gases, oxygen, air, and nitrogen, on cellular effects of monocytes and osteoclasts in both normoxia and hypoxia. And the following slides just show some highlight data from these studies. So first, looking at the panel on the left, first thing we noticed was that osteoclasts, which are labelled OC here, are much more sensitive to nanobubble-induced cell death than the monocytes from which they derive, and this is regardless of the gas with which the nanobubbles are loaded. So this effect is quantified in the top 2 graphs in this panel. You can see here the highest dose of nanobubbles in monocytes after 3-day exposure only induces a 2-fold increase in cell death, whereas the same dose given to osteoclasts for only 24 hours induces a 20-fold increase in cell death. The 2 graphs below this are the relative number of cells present in these 2 situations, and as you'd expect with the osteoclasts, where you're seeing increased cell death, we see reduced osteoclast numbers, although there's no significant difference between the gas-loaded bubbles. Similarly, in monocytes, at the highest dose, we see a reduction in monocyte number, with no difference in magnitude of effect between the gas-loaded bubbles. And at the lower doses, we see a gas-dependent effect, although in this instance, it's that the nitrogen-loaded bubbles suppress proliferation of the monocytes, whereas the air and oxygen-loaded nanobubbles do not. However, we also see gas-dependent effects on hypoxia signaling. So if we look at the right panel now, we're looking at induced expression of the hypoxia-inducible transcription factor HIF1 alpha in hypoxic conditions. So in normoxic conditions, HIF is not present. In hypoxia, the protein is stabilized. And if we expose the cells to the different gas-loaded nanobubbles at the same time as hypoxia, we see that the oxygen-loaded nanobubbles, the amount of HIF that is produced is much lower. And this suggests that the hypoxia signaling is being at least partially inhibited by oxygen, by having oxygen in the nanobubbles, whereas nitrogen, if anything, the hypoxic is worse. And we see a similar differential between nitrogen and oxygen-loaded nanobubbles in osteoclasts. However, the biggest effect that we see is when we look at the effect on osteoclast differentiation. So as I mentioned previously, osteoclasts form by fusion of monocytes, and they are considered osteoclasts when they are TRAC-positive and have 3 nuclei. But there is a pre-osteoclast stage, which is in the top panels on the left here, where they do not have 3 nuclei, but they are starting to morphologically have the fried egg morphology that osteoclasts exhibit, despite only having 1 or 2 nuclei. And if during the differentiation process we expose the monocytes osteoclast mixture to the nanobubbles, we see that oxygen-loaded nanobubbles reduce the number of pre-osteoclasts formed at low doses. At high doses, there's just a general suppressive effect of the nanobubbles themselves. The later stage of differentiation, these pre-osteoclasts fuse further to form multinucleated osteoclasts with 3 or more nuclei. And in this state, this is a 24-hour exposure, so during this time the osteoclasts fuse, but in the presence of the nanobubbles, there is even greater fusion with oxygen-loaded nanobubbles than nitrogen-loaded nanobubbles at low doses, and again, high doses are suppressive. But actually, the overall story here, which I haven't photographed for in this talk, is that if we expose the cells to low-dose nanobubbles for the duration of the differentiation period, it is this effect, which is the overriding effect. So generally, continuous exposure to low-dose oxygen nanobubbles during osteoclast differentiation reduces the final number of osteoclasts that are formed. Moving on to the right-hand panel, the most striking effect we see with gas-dependent differences is in osteoclast fusion under hypoxic conditions. So if we look at the left-hand of the two image panels, minus 24 hours is our starting point, so we have pre-osteoclasts. 24 hours later in normoxia, most of the cells are fused and we have large multinucleated osteoclasts. If this fusion occurs in hypoxia, if this process occurs in hypoxia, the fusion process does not occur. But if the cells are additionally exposed to the oxygen-loaded nanobubbles, then they are able to fuse to a similar degree to they do in normoxic conditions. This is quantified here. So fusion in normoxia, reduced fusion in hypoxia, and at the low doses of the nanobubbles, the oxygen-loaded nanobubbles rescue the reduced osteoclast fusion under hypoxic conditions, whereas if anything, the nitrogen-loaded nanobubbles make it worse. So in summary, we've shown that high-dose nanobubbles have a general inhibitory effect on monocytes and osteoclasts, which is not dependent on the gas with which they are loaded. However, it's unlikely that in the in vivo situation, the cells would be exposed to this dose of nanobubbles. At lower doses of nanobubbles, we are seeing gas-dependent effects, and it's more likely that these are the sort of doses that the cells would be exposed to in vivo. So we've shown that the oxygen-loaded nanobubbles are able to reduce HIF expression in hypoxic monocytes, so that's saying they can affect hypoxia signaling and reduce hypoxia signaling in vitro. We've shown that the oxygen-loaded nanobubbles also prevent pre-osteoclast formation in normoxia, and that this is extended so that if they are exposed to this for the duration of differentiation, the oxygen-loaded nanobubbles reduce the final number of osteoclasts that are formed. However, the same oxygen-loaded nanobubbles protect against reduced osteoclast fusion under hypoxic conditions, and we are still following up on this result. In the future, we need to identify the molecular pathways that are involved in the gas-dependent effects of nanobubbles in both monocytes and osteoclasts. We also want to ask whether the nanobubble composition can be modified to minimize the general inhibitory effects seen at high doses, although there are questions about whether this is necessary, because it's unlikely that this dose of nanobubble would be seen in vivo, or even desirable if the effect on cell death is only really evident with osteoclasts, which have a poor prognostic effect on rheumatoid arthritis. We're performing similar studies in other cell types, so Tom Law is a PhD student in the lab who's looking at synovial fibroblasts and endothelial cells. On the back of the data we've generated here with the osteoclasts, a collaboration has been initiated with other groups who have started to perform studies on oxygen-loaded microbubbles, which are a larger version of the nanobubbles we've been using, and have also been looking at effects on osteoclasts in bone. The clinical study that I mentioned as objective 2 of the overall project has been delayed due to COVID, although hopefully should be starting imminently. And if we can show that orally delivered nanobubbles can inhibit hypoxia pathways in vivo, and hypoxia pathways are known to be associated with multimorbidities of aging, this could be a valuable intervention that's capable of increasing the health span in not just rheumatoid arthritis, but other aging-related conditions. Thank you very much.
Thank you very much, Helen. And I think it's really exciting to see this platform and think about the wide-reaching applications it could have, particularly in this field. Now, we're going to carry on and hopefully get some time for questions at the end. So in our all-star female cast for our short talks, I'd like to hand the virtual floor over to Claire West. Claire West joins us today as a postdoctoral researcher working for UK Spine across the Centre for Medicine's Discovery and Open Targets EBI-EMBL. And Claire's here to talk to us today about her bioinformatics work with Target Age. So Claire, the floor is yours.
Okay, great. Thank you very much, Bev. And can you see my slides okay? I'm going to assume yes. So yeah, thank you very much for having me here today. My name is Claire West. I'm a postdoc based at the University of Oxford and working with open targets and CMD and UK Spine. And the project I'm going to talk about today is our project. We've been doing it since January last year, and we're looking to computationally identify potential targets for age-related multimorbidity. So just to reiterate the problem that's already been set out earlier in the conference, the need for developing new therapeutics for healthy aging is becoming more apparent as populations age. So in the UK and globally, life expectancy is increasing, which is great, but the number of years spent in healthy life is not increasing at the same pace. And as a result, people are spending an increasing amount of time in their late life living with morbidities. And for more than half of people over 65, this is more than one long-term condition, and this is what we call multimorbidity. And for these patients, they're often taking multiple medications to control these different diseases. So the traditional paradigm of studying diseases individually, developing a single drug for a single disease, is probably not best serving these patients who are suffering from multiple diseases and taking multiple drugs. So aging is sometimes or often defined as time-dependent decline. And this begins as subclinical physiological changes in the body. It continues to systemic changes in the body and eventually culminates in age-related diseases that are defined as different diseases and eventually leads to frailty and multimorbidity. And the rate at which this decline occurs relative to chronological age varies a lot between individuals, and there is evidence that we can modulate this through interventions. So the pathways of aging itself are thought to contribute to this decline and to some extent to the development of these age-related diseases. And so our hypothesis is that rather than approaching each individual disease mechanism to try and develop a drug, perhaps we can identify targets at the intersection of multiple age-related diseases and the aging process itself. And this could be a more effective way to treat and prevent age-related morbidities. So this is our idea, and this is a great thing to try and pursue, but the real challenge is to identify what those targets are and determine whether or not those are really going to be good therapeutic opportunities. So we're taking a computational approach to this problem. So we're saying, for all of the huge amount of biological data that's out there, how can we use that data to try and find these targets? And there's 8 parts to this process. I'm just going to really briefly talk about each part. The first thing we need to do is look at the data that we've got and determine how we're going to use it to answer the question. So I'm working with Open Targets. For those of you who don't know Open Targets, they're a public-private partnership. It's part of EMBL EBI in Cambridge, in Hingston, Cambridge. And they are a partnership that systematically identifies targets for different diseases. They have 2 main resources. The first resource is Open Targets platform. And this brings together a really vast amount of public domain data, and it uses that data as evidence to draw associations between targets and diseases. So this is the starting point for the project. They also have Open Targets genetics, and this aggregates lots of different GWAS data, and it performs analyses on that data to identify causal genes. And this then feeds into the Open Targets platform. They also have a growing validation lab. This is in collaboration with the Wellcome Sanger Institute. And here they perform experimental and computational work to probe these target disease relationships that are coming out of the platform and Open Targets genetics. So we've already got this really amazing resource and ways that we can ask questions of target disease relationships. We also bring in extra information relevant to aging. So for example, we bring in databases like CellAge and GenAge, which are manually curated databases of genes that already have a link to aging. So either they are known to modulate longevity in animal models, or they affect cellular senescence in cell models. So by combining Open Targets and other databases relating to aging, we are hoping to use this data to answer these questions. The next stage is identifying what's the scope. So what diseases are we interested in? And we have curated a list of age-related diseases and traits that we are interested in. This includes diseases like type 2 diabetes, osteoarthritis, sarcopenia. It also includes traits such as grip strength and frailty. And we define these diseases using EFO, which is a hierarchical ontology. And this allows us to define the different sub-levels that we're interested in, and means that we can aggregate data that has been mapped to more specific subtypes of a disease, as well as those that have been mapped to the broader disease. So in this example, I'm showing a section of the ontology relating to arthritis, and we're interested in evidence that is mapped to osteoarthritis and rheumatoid arthritis, but we're also interested in evidence that's mapped to more specific subtypes of osteoarthritis, such as osteoarthritis of the hip or of the knee. And using this approach, it means that we can get as much information as we possibly can about these diseases. So we have expertise through open targets in genetics, and we also have at the CMD expertise in enabling targets that have been historically underexplored. And because of that, we're particularly interested in genetic links to diseases. And so we're interested in identifying targets that are genetically associated with multiple age-related diseases. And the way that we go about finding those is we collate all of the GWAS studies that have been performed for disease 1 or disease 2, or all of our diseases of interest. So in this diagram, the grey lines are representing different GWAS studies for slightly different traits relating to disease 1. The blue lines are GWAS studies relating to different traits related to disease 2. And the yellow circles represent loci that have been associated with these diseases through these GWAS studies. So the first thing we need to do is identify, once we've got together all of these GWAS studies, how many independent genetic signals are we looking at? And this means we're seeing, of all of these genetic hits from the different GWAS, how many of those are likely to be caused by the same causal variant. And this allows us to draw these black lines between associations that we think might be related. And we do that using colocalization or LD expansion. So once we have this, we have an idea of how many unique independent genetic associations there are for each disease. And then we can look at how many associations are shared between diseases, represented by these dashed lines. And once we have this, we can see to what extent the genetic signals for these diseases are distinct to each disease and to what extent they are shared between diseases. And these shared associations may indicate some extent of shared mechanisms that might underlie both diseases. And we can kind of quantify which diseases are more genetically related or more further apart. And once we've identified these genetic associations that are shared between diseases, we are looking at what are the causal genes that might be behind these associations. We want to know, do they co-localize with a quantitative trait? So do they have an effect on the abundance of a protein or expression of a gene? And this is linking it back to the biology. And when we have that, we can also go one step further and perform Mendelian randomization, which can give us an idea of whether that change in the abundance of the protein or the gene might be causal for the disease. And is that effect the same for both diseases? So is an increase or decrease in the protein have the same effect on both diseases? And if we can identify those genetic associations that have an effect on a protein target, and that might be causal for both diseases, then we have quite strong genetic evidence that this could be a potential therapeutic opportunity. So in addition to genetic information, we're also interested in the expression of these targets. So for example, if we have target A that is associated with disease 1 and disease 2, we want to know what is the baseline expression of this target in different tissues. Are those tissues relevant to our diseases? And to connect that back to aging, we want to ask, are these targets involved in age-associated expression signatures? So in other words, is there evidence that this target either tends to increase or decrease expression with age? And therefore, that might suggest that it's involved in the aging process. Another way to link these targets back to the aging process is to identify whether or not they fall in the pathways of aging. So these are the hallmarks that have been mentioned already in the conference. These are hallmarks pathways such as cellular senescence, loss of proteostasis, DNA damage and repair. And we have mapped these pathways to Go terms, which are used to annotate genes. And when we do that, we can see which targets fall within these pathways, which gives us kind of an extra link to aging. So in addition to identifying which targets are associated with multiple age-related diseases and which ones are related to the aging process, we're also interested in whether these represent good drug development opportunities. And so the kind of information we want to make this decision is there already an existing link to aging? Is this already known to relate to aging based on the cell models or animal models? Is there clinical precedence for this target? Is there a drug already for one of our diseases or a different disease that we could repurpose? Is there a drug in clinical trial? is there a chemical compound that is known to bind to this target? We also want to know how novel this target is, so we can look in the literature to see how many times is this target co-mentioned with our diseases of interest or with aging. And this gives us an indication of how strongly that target has already been associated with these diseases and how novel it would be in that perspective. We're also interested in whether the target is a member of the druggable genome, which suggests that it could be a good target. Is there a PDB structure available for this target that could help us decide whether or not it's good? Are there any chemical probes or target enabling packages for this target? Is there any information on safety and toxicity that we can take into account that's available? And finally, is there any internal knowledge among our UK spine partners that might help us make a decision on whether or not this is going to be a good target for us to take forward? So once we have all this information, we need to do a triage to decide whether or not we're going to take these targets forward. And to do this, we have these target selection meetings that brings together different partners in UK spine. And we then discuss the methodology and we triage particular targets. And this is really about harnessing the different expertise that we have in the network. So for example, Open Targets has a lot of expertise in genetics and in computational biology. The CMD is very strong on structural biology. The Drug Discovery Unit in Dundee and the Medicines Discovery Catapult in Manchester are, of course, very experienced in drug discovery. And Birmingham has a really deep knowledge on aging biology and clinical trials. And so through these meetings, we can really bring together the different capabilities and expertise and decide which targets are going to be good for us to take forward and who might be best placed to take that forward and what we might need to do to facilitate the development of a drug for that target. So really what this is doing is bringing together a huge range of annotations for targets that we think might be involved in age-related diseases. And there's a broad range of targets that we're pulling out of this. So some of them, for example, might already have a really strong link to aging. They might already have a lot of precedence. Some might represent quite good drug repurposing opportunities. Others might be extremely novel. They might not have much validation information already, but show promise through their genetics. And so there's a big range of the targets, and there's also a range in the interests because different people are interested in different levels of novelty and precedence. So to facilitate the exploration and the prioritization of targets, we developed a web application. It looks something like this. And this allows us to explore the targets. We can see for each target all of the information that I've been talking about so far. So to what extent is it genetically associated with each of our diseases and traits of interest? Is it already linked to aging? Does it have an effect on longevity or senescence? Is there clinical precedence? Is it in a hallmark of aging? And then information about the target itself, a description, PDB structure if there's one available, all this kind of information is presented together. And at the moment we're using this within UK Spine, but we hope that this will become a publicly available resource through Open Targets in the future. So just to summarize, this is the process that we're taking. This is a summary of our approach. We are looking for shared genetic associations between different age-related diseases and traits. We're interested if they have an effect on protein or RNA expression in relevant tissues. We're looking for evidence that it's causal for these diseases in the same direction. We want to know how they relate to aging. So are they in an expression signature? Are they in a pathway of aging? Do they have an effect on longevity or cellular senescence? And finally, we're interested in how novel are these targets? How tractable are they? And do any of them represent good therapeutic opportunities? So thank you very much to everyone who's contributed to this project. There's so many of you at the University of Oxford, Open Targets and UK Spine. So thank you very much to everyone, and thank you for listening.
Thank you so much, Claire. Super presentation, showing a huge range that we've covered this afternoon with you lovely ladies. We've got a couple of minutes, so if I could get Helen and Sartomi to join back in. Some questions have been coming through and we'll attempt to do as many as we can. So let's have a look. The first question I think is for Sartomi. Now, it's quite a long one. I'm going to give it a go. Oh my. Navitoclax leads to back-dependent apoptosis. Are there any important senescent cell types found in vivo or in human disease that you might expect not to be effectively killed by this treatment? Or It's not quite finished yet. Or conversely, do you know what cell types / subtypes had been depleted in your treated mice versus the control mice? Oh, I've done it. I've got to the end. So tell me.
Now it's my turn to answer. Yeah. Yes. So senescence cells, we call senescence cells, but the exact nature of senescent phenotype is different depending on cell types. So muscle cells don't tend to develop same kind of expression changes as say fibroblast. So those senolytic has cell type specificity. So that's Navitoclax is good for particular cells, but not others. However, what we see in vivo is that nonetheless, overall, mouse organism level of heredity and other neuromuscular function, cognitive function is improved. So this also suggests it has a paracline effect. So not just a single cell type affecting whole organism aging phenotype, but maybe subset of senescent cells is enough to cause systemic high inflammatory state. And that's one potential idea. And co-targeting of drugs, so depending on target of senolytics, specificity of cell types are different. So another senolytic is disatinib quercetin. So these two drugs are combination target more type of cells. So that's another approach. And then, yeah, what is?
No, I think that's a super answer, Sartomi. It was by virtue of the lady that asked it. A long question is her specialty, and I mean that in the most wonderful way. I'm going to try and get through a question for all of you before we finish the session. So my next question is for Helen, and Helen, slightly shorter. What might a nano-based treatment look like clinically? Would this be a regular invasive treatment or an intensive therapy to repair damage for the medium or longer term?
So the strategy we're looking at the moment is an oral intervention. So they've developed it's basically a drink so it can be taken orally, which is what was done in the xenograft study and with the male cyclist. It was delivered as a drink. We would anticipate this would be a long-term treatment because we don't think the dose would be high enough to dramatically reset the hypoxia pathway. But potentially, if you gave people this drink periodically from when they had early disease, it might reduce the level of hypoxia over the long term, which would reduce one of the driving forces behind disease progression. So it might slow disease, improve disease symptoms in the long term. Obviously, it's quite an early stage. We need to know whether this actually penetrates the hypoxic tissues, although these preliminary studies look like it might. So like the mice, the reduction in HIF we saw was in the tumour tissue only 30 minutes after oral administration of the nanobubbles. So there is evidence that it could do that. Obviously, we now need to look in humans, which is what the clinical aspect of our study is about. But we would anticipate it was a long-term, not quite preventative, but reducing effect rather than a specific hard-hitting treatment.
I think this is so fascinating because you can see a model whereby it's very easy to deliver a drink, an oral-based treatment to many patients. And we know that compliance is something that we often hear about being an issue for people actually adhering to the medicines that can help them. So thank you very much, Helen. And just last but by no means least, Claire, I have a question for you. So for expression analysis, knowing what meaningful levels of expression are in, I don't know what that is, that word. Expression can be very tricky, either at RNA or protein level. What method are you using to look at expression, and what controls do you have to know if you're looking at a meaningful level of expression?
Yeah, thanks for the question. This is one of the benefits of working with open targets and envoys that, sorry, my fan's a bit loud. One of the benefits of working with envoy is that these questions are answered in a systematic level when they're put into the resources. So when open targets is considering expression information to decide PPTLs and things like that, PPTLs, these kind of cutoffs are determined at that level. It's combining lots of different experiments, and the expression is also using expression patterns, I think. So these kind of decisions are made by these big teams that curate these resources for us, and make sure that all of the information is being integrated in a sensible way. So it makes it easier when I'm doing my stuff, because all of these decisions have already been made. And then when we're looking at individual targets that come out of it, we can, so I'm just going to check, I do have some.
I think we're just going over to headphone mode. Oh, no.
Sorry about this. Yeah, so when we're looking at individual targets, of course, we can look at these on an individual level and see what's the evidence that's going into our decision making. But when we're doing at the large scale, when we're looking genome-wide, these decisions and cutoffs are being determined by the resources that we're using.
I think that's a good point to pick up on about the resources. I think we're seeing a growing bank of resources, as we talked about yesterday. And Claire, I mean, you look quite widely at pulling in everything from biobank, from open targets, and much wider than that. And I think that's part of the power of consortia such as these. So thank you very much. Now, I realise I've gone 2 minutes past my allotted time, but I'd just like to say a huge thank you to the ladies for joining me for this session, and I hope everybody listening has enjoyed it. We're going to take a short break till 4:15 now, where we're joined for our panel session. I'd really like to say, once you have stretched your legs, had a cup of tea, we do have a networking platform on Hopin, and we're all trying ever so hard this week to get into the technological age of virtual conferences. You can use that platform to go and say hi to people that you might have done so over a cup of tea. So please do that, and we'll see you back for our last panel session, chaired by Martino Picardo, our returning excellent chair, in about 45 minutes or so. So thank you very much, everybody. Bye for now.